Proportional meta-analysis in R statistics and generation of forest plot and funnel plot. Proportional meta-analysis is a single group meta-analysis which is different from pairwise meta-analysis. In pairwise meta-analysis, there is two groups, one intervention or exposure, and the other one is a reference or unexposed group. In a single group meta-analysis, only one group of data are used. So what is proportional meta-analysis? It is a statistical method that combines results from several studies. Those results or data extracted from multiple independent studies. The effect size estimated or measured in this analysis can be percentage, proportion, rate, or frequency. So the proportional meta-analysis will calculate the pooled overall estimates of the proportion or the rate from those several independent studies addressing specific scientific, medical, or clinical question. So any binary data that is reported in percentage or rate or proportion can be included in the meta-proportional analysis. The method is used commonly for the analysis of the prevalence or incidence of condition or disease in population. The meta-analysis also will assess the degree of variability or inconsistency among the results from those several independent studies by performing Q statistics, which is based on chi-square, and a p-value is generated, and an I-squared, which is represented in percentage. An I-squared of 0% indicates that there is no heterogeneity between the results from those several independent studies, and 100% I-squared value indicates maximum heterogeneity. The proportional meta-analysis also generates a forest plot. A forest plot is a graph that displays the summary of the outcome from the meta-analysis. It will include in it the proportion or the rate, the 95% confidence interval, could be a p-value, a weighted percentage. So the graph or the plot is made up of three sections: statistical summary, diagrammatic representation of the statistical summary, and the third part, which is the overall estimate of the effect size or pooled effect size, which in this case the proportion or the rate, and the test of heterogeneity with I-squared value and the p-value. The proportional meta-analysis will also produce a funnel plot to test and examine the publication bias, and the plot is a scatter plot. The funnel plot is a scatter plot in which in the x-axis the estimate of the effect size, which is the proportion or the rate or the percentage for each specific study, is plotted on the x-axis, and on the y-axis is the standard error, and the plot is in a shape of a funnel, and the distribution of the effect size for each studies under the funnel will indicate whether there is bias or no bias in publication. To perform proportional meta-analysis in R, launch the R Studio, and then also load the meta package. If you have not installed the meta package, you need the meta package for performing meta-analysis. I've already had installed the package, so I'm going to load it into the RStudio by typing library, open a bracket, and then type meta library, open a bracket, meta, and hit enter, and the meta package is loaded into the R. To load the file that contains the data for meta proportion analysis, click on File, scroll down to Import Data Set, and then select from SPSS as the file is saved in a format of SPSS. If the file is saved in another format such as Excel or text or SAS or Stata, then choose the right options. I will click on the file from SPSS, and then in the new window, browse to select the file. Once the file is located, select the file and click on Open, then click on Import, and the data is loaded into R. As shown here in the top section in the table, there are 12 independent studies labeled from 1 to 12 with a label of study ID named for that variable, and a second variable which is the event for specific condition, and then the total number of participants or recruited for the study. So to perform meta-analysis, proportional meta-analysis on these 12 studies, in the section here, let's generate an object and call it X, and then use the assignment operator and type meta proportion, as we are going to perform a proportional meta-analysis. So this is the command for single group meta-analysis, which is meta proportion, as I have here only one set of data, and in between the bracket type data equal to and the name of the file or the object that has been loaded into the R, which is this one. Then insert comma, and after that insert the variable that contains the event, which is labeled as event, then insert comma, and then the variable that have the total number, which is total, in this case capital letter total, then insert comma, and after that type method, and I'm going to select here the method which is inverse between two quotation mark and inserting an I symbol for inverse. Then after that insert a comma, and then for the summary type SM equal, then quotation, and then P row for proportion row, then followed by comma, and then to also insert the study ID for each individual study. So we need to use the variable which is labeled study ID by typing stud lab or labels equal to, and then study ID, as it is case sensitive in R, so it has to be entered as it's recorded in the table. Then insert comma. Here I'm going to use the fixed effects model by typing first random equal to F for false, and then fixed comma, and then fixed equal to t, which is true. So here what I have decided is that to use the fixed effect model and not the random effects model. If I want to swap, if I want a random effects model, then I will type random equal to true and fixed equal to false. But I'm going in this video use the fixed effect model. So this is the command to perform meta-analysis, proportional meta-analysis. The way to do that is to generate an object, call it x, then use the function which is meta proportion, open a bracket, type data, what is the name of the data, which is this one, then comma, then insert the first variable which is event, then the next variable which is total, then the method which is an inverse, and the summary which is the proportion row, and a study label is inserted also with a fixed effects model chosen. If I hit enter to view the proportional meta-analysis, just type x. As shown here, there are number of studies shows that there are k equal to 12, and number of observations 1212, and the number of event 426, and the model used here is the fixed, it's called here common effect model, and the proportion is calculated. This is the overall or pooled proportion, which is 0.34, and the 95% confidence interval, and the proportion is within the lower and the upper limits for the 95% CI. Also, a heterogeneity test was performed and an I-squared is generated, which equals to 70%, and the 95% confidence interval for that value is also generated, and the test of heterogeneity performed using the Q statistics with the value produced, and degree of freedom and a p-value less than 0.05. To draw a forest plot, just type forest and then open a bracket, insert the object which is x, and if we want to draw a forest based on a layout by revaman, then type layout quotation mark and then revaman and hit enter. A forest plot is generated in the format of revaman as shown here: the study IDs, the events, the total, the weight, the model effect which is the fixed, and the 95% confidence interval, the diagrammatic representation, the scale, and the overall values. However, if we want to do a forest plot and draw a basic forest plot, edit the plot as we wish, then close that plot, and in the section here type forest, open up brackets and then type x, and a basic forest plot is generated. As shown from this plot, the study ID is inserted from study 1 to 12. The events are added here, and the total for all the 12 studies are calculated. However, the total for the events is missing here, and in the middle section is the diagrammatic representation, and the proportion is given here, and the 95% confidence interval with the weight percentage, and an overall effect size which is a proportion, and a heterogeneity test. If we want to add the total number of events to the plot, close the plot, and in the section here type forest, open a bracket, x comma, and then type pooled events equal t for true, and hit enter, and then click on zoom again. As shown here, the total event is added here. To add a label here for the diagrammatic representation, close the plot and then copy the command for the forest, insert it here, and then comma after the true, and then type sm summary label equal to, and let's give it a name which is proportion. The proportion word has to be between two quotations marks, so type proportion and then hit enter. Click on zoom again. As shown here above the diagrammatic representation of the forest plot, the proportion label is added. To change the color for the squares and the diamond and the vertical line here, the square here represent the proportion for each individual study. The diamond represent the overall or pooled proportion from the proportional meta-analysis, and the vertical line represent the overall proportion. To change the color for the square symbols here, close the plot and in the command section copy the command for forest and then insert that here, and then insert a comma, and to change the color of the square symbol type col dot square equal to, and let's give it a color of red, and then hit enter. Click on zoom, and as shown here, the square the effect size for the proportion, which is the square, is changed into a red color. To change the color of the diamond into a black color, exit the plot, copy the previous command and insert that here, and then after the quotation insert a comma. To change the color of the diamond, type col dot diamond, select the color dot diamond, and then equal sign and quotation mark and type black, and then hit enter. And as shown here, if we click on zoom, the diamond color is changed into black. To change the color of the vertical line, close the plot, copy the previous command, insert that in this place here, and then insert a comma, and then type col dot common and open a quotation mark, and let's color code it blue and hit enter. And if we click on zoom, we can see that the vertical line is colored blue. Could also change the scale in this ruler here from 0.1 to 0.7, and the way to do that is to close the plot, copy the previous command, copy and then insert here, and then after the quotation insert a comma, and then type x limits x lim equal to, and then insert c. The lower value will be 0.1 and the upper one will be 0.7. If we hit enter and then click in zoom, as shown here. The scale is redrawn from 0.1 to 0.7. So this is the forest plot. From this forest plot we can see that there are three sections: the statistical summaries, the diagrammatic representation, and the overall statistics. Here, as shown, the studies are inserted from study 1 to 12 with the events in each study and the total of participants in each study, and the proportion is calculated for each independent study with a 95% confidence interval and a weight percentage, which reflects the sample size, as higher samples that have a larger number of participants will have a higher weight, as the weight can influence the meta-analysis. The second section here, which is a diagrammatic representation of the descriptive statistics. The red square here represent the proportion for each individual study, and the size reflect the sample size. The size of the square reflects the sample size. The horizontal line represent the 95% confidence interval with the lower and the upper limits for it. The vertical blue line is the represent the effect, overall effect size or overall proportion, which is 0.34. And the third section is the overall statistical analysis, or statistical analysis for the overall estimate effect size and heterogeneity. As shown here, the overall proportion is 0.34 and 95% confidence interval is calculated. And for heterogeneity, the I-squared is 70% and the p-value is less than 0.05. So this forest plot shows that the 12 studies, or the data from the 12 studies, were pooled, and the overall estimate of the effect size, which is the proportion, is calculated. To draw a funnel plot to check for publication bias, close this plot, and in the section here type funnel, open a bracket, and then insert the object, which is X, and hit enter. Click on zoom. A funnel plot is generated as shown here. This is a scatter plot with the x-axis represent the proportion for each individual study as indicated by gray circles, and on the y-axis is the standard error, and the shape of the plot is similar to a funnel. The line in the middle here represent the overall proportion, which is calculated to be about 0.34. And for lack of publication bias, the distribution of the effect size, which is the proportion for the studies, has to be equally distributed in both sides of this line. So it has to have symmetry in the distribution. From this funnel plot, the publication bias can be assessed, and to further validate the finding of the funnel plot, then Eager's regression test can be performed. So this is the way to perform proportional meta-analysis in R statistics and generating a funnel plot and a forest plot.